Bismillahirrahmanirrahim. Today I will talk about downstream processing. So it is the most important process of biochemical engineering or industrial biotechnology. So downstream processing, the various procedures involved in the actual recovery of useful products after fermentation or any other process together constitute downstream processing. After fermentation, we have issued or targeted product. So these product, there are many impurities in our product. So we isolate our targeted product and purify that product, and it is called downstream processing. It is a very important step in the manufacture of different products in pharmaceutical industries such as antibiotics, hormone, antibodies, vaccines, enzyme, food industry, and so on. So in addition, the product is either present in the cell, in the medium, or both. So our product is either present, it is present inside the cell or present in the medium. So we extracted those products from the medium or the cell. This is upstream processing. First of all, we insert our target product in vector and we pump a colonies inside a host cell, and the targeted product is achieved in a very small quantities. After that, we perform fermentation and the product is produced in a large quantities. After fermentation, we perform downstream processing, in which cell lysis, purification, and after purification we achieved our targeted product. Now various steps in downstream processing. The first step is cell disruption, removal of insolubles, product isolation, product purification, and product polishing. We will discuss one by one. The first step is cell disruption. Disruption of microbial cell is usually difficult because of their small size, rigid cell walls, and high osmotic pressure inside the cells. Disruption of cell is generally achieved by mechanical lysis or drying. Mechanical cell disruption. This involves the use of sonication, homogenizer, and ultrasound. So these are the steps, mechanical cell disruption steps. Now the other one is drying. It involves the drying of cells by adding the cells into a huge amount of cold acetone and extracted using buffer or salt solution. Now lysis of microbial cell may be achieved by chemical means such as salt, osmotic shock, freezing, or lytic enzymes, for example, lysozymes. Now the second step is product isolation. It is the removal of those components whose properties vary markedly from that of the desired product. For most product, water is the chief. Impurities in isolation step are designed to remove most of it, reducing the volume of material to be handled and concentrating the product. The process of isolation is achieved by solvent extraction, adsorption, ultrafiltration, and precipitation. Now the other step is purification of the product. It aims at recovery of the product in a highly purified state. Purification is achieved by the following procedure. Crystallization. This is used for the low molecular mass compound like antibiotics. Chromatographic method. It is generally used for the purification of low molecular mass compound from mixture of similar molecules, for example, antibiotics and macromolecules such as enzymes. Drying. So So drying is the most important step in downstream processing, which makes the product suitable for handling and storage. The most frequent approaches of drying are vacuum drying, spray drying, and freeze drying. So this is all about of our topic downstream processing. Thank you.